Hello, I'm Nathalie Reyes, an undergraduate student from Florida Gulf Coast University. I will be presenting my research that was supervised by Dr. Lyndsay Rhodes of analyzing fibronectin gene expression in native pancreatic and pancreatic adenocarcinoma samples. In the United States, pancreatic cancer is the fourth leading cause of cancer death in men and women. It accounts for 7% of all cancer cases and has a 5-year survival rate of 10%. What this means is that in 100 patients, only 10 of them will survive from this cancer. Pancreatic ductal adenocarcinoma, or commonly referred to as PDAC, is a type of pancreatic cancer that forms in the pancreatic duct, where the acinar cells and ductal cells reside, causing patients to experience a lack of appetite, weight loss, nausea, or no symptoms. The extracellular matrix, or the ECM, is a complex network made up of micromolecules like proteins and carbohydrates, which is located in between the cells. In PDAC-bearing pancreas, the pancreatic stellate cells become increasingly activated, resulting in the accumulation of ECM proteins. This leads to a stroma-rich environment and increase in tissue stiffness. The ECM environment and its proteins promotes the hallmarks of cancer. Fibronectin is a glycoprotein that provides structural integrity in the ECM. It also binds to membrane receptor proteins to carry out signaling pathways. It is a normal component in the pancreas, but in PDAC-bearing pancreas, fibronectin takes advantages of its abilities to promote tumor cell survival, cell proliferation, and cell migration. It is still unclear why and how fibronectin affects patients with PDAC. Therefore, we are conducting this study to determine if the expression of the fibronectin family of ECM proteins is correlated to decreased survival in PDAC patients. To determine how fibronectin affects PDAC patients, we are utilizing XENA and GEPIA2 to visually and statistically compare the level of fibronectin expression between human native and tumor pancreatic tissue samples. Many of the analyses consist of samples that are half tumor and half native. Through this study, we have learned that the level of fibronectin expression is higher in tumor tissues than in native tissues, as shown in Figure 1. Also, through Figure 2, 3, and 4, we notice that if fibronectin expression increases, the probability of overall survival decreases, and then the probability of reoccurrence and disease progression will increase. We also see in Figure 5 that fibronectin expression increases as patients progress through the stages. The next steps of this research will be to determine if expression of the different regions of the fibronectin gene affects PDAC patients' overall survival. Thank you for listening to my presentation for FURC 2021. I would also like to thank FGCU, the Honors College, and my mentor, Dr. Lyndsay Rhodes. Thank you.